think one of the themes that really stood out for me was a lot of the work that's going on in cancer, specifically in cancer resistance, and how single-cell technology can really illuminate that, not only for scientists, but also for clinicians.
Well, I really enjoyed Prisca Liberali's talk this morning, where she was talking about gut development. And I was thinking about how we could apply some of the principles that I've been seeing in development to look at our questions, which are cancer related, so completely different. But there are some ideas that are coming up.
Yeah, I mean, I think what we're seeing is the ever expanding power of this kind of approach that we've been using, that when you look at the single cell level, there's just so much light that you shed on phenomena. So what we've been seeing is us looking at cancer, but also studying organoids and different diseases, just more and more problems that can be tackled by this approach.
Yeah, I think we've seen several talks so far. For example, I'm just coming off from Ellen Rothenberg's talk, where she has shown an expert, genome-wide perturbation screen of T-cell specification signals in the mouse. Earlier at the meeting, Cole Trapnell from the University of Washington had a really beautiful study where he's really started to do organismal level genetic screens with single cell readouts, where he has come in and he has knocked out 30 different, transcriptional or gene expression regulators in zebrafish. And he is looking at the development of the whole zebrafish embryo with these single cell methods. So to be able to do that depth of analyses, where you're not just you're not just perturbing one property of an organism or a tissue and looking at a given cell type, you're perturbing something at the level of the whole organism. You're looking at all cell types at once across the whole body. That scale and insight is really interesting. I think it's quite new so you can look at whole parts in different ways, right? It's people have been looking at whole bodies at superficial levels for years, right? But to say that we are going to take an entire organism and create single cell level atlases of the healthy organism, or the organism under perturbations or under different environmental conditions or diseases, is, I think, completely new. And it's really fueled by these technological advances in single cell biology over the past several years. And I think, it's really great to be, I think one of the highlights of this meeting again is that, you have lots of the pioneers in the field attending, and you're getting to actually find out about these tectonic shifts in technology and single cell biology in real time.
I think we are at the moment where we are generating lots of data from using different technologies, and really all of this is key to understand what a cell is, but also how it interacts with its environment. And we are at the moment of where it's super exciting to be in this community, and from the talks we can see more and more modalities kind of coming, even those that will allow us to understand the mechanistic forces and how it has an effect on the cell. And the other thing is how the computational analysis and new machine learning tools are essential to really make sense of all this data that is being generated. So yeah, overall I feel it's quite an exciting time to be, and we could have seen this in the presentations.